Well, Biology Part 222, calculating LLA frequencies, and we've got the scale for Hott-Levyenberg method. We've got the frequencies of CLLA, and then we've got the formulas. There are labels for the frequencies, and we accordingly monitor them. The transient and balanced polymorphism. Polymorphism is said to occur when individuals of one species in the same population exist in two or more distinct forms. These different forms are called morphs and must be phenotypic variations of the same character. For example, normal and melanic moths are alternative morphs for body color. The transient polymorphism arises when there is strong selection pressure against one of the morphs so that it will eventually disappear from the population. In balanced polymorphism, on the other hand, the selection acts to maintain the different morphs indefinitely. The Hardy-Wennberg method can be applied to show that balanced polymorphism is likely to arise for characteristics controlled by a single pair of alleles. Whenever individuals with the heterozygous genotype are at a selective advantage compared with the homozygous dominant and the homozygous recessive individuals. An important example concerns the human disease sickle cell anemia. The disease is caused by the recessive mutant allele, which codes for an abnormal hemoglobin S. The homozygous recessive individuals, SS, have red blood cells which contains only hemoglobin S. This becomes insoluble at the low levels of dissolved oxygen, causing a change in the shape. The deformed red cells block the body capillaries, resulting in tissue damage, so that the affected individuals rarely survive to reproductive age. The heterozygous individuals, SS, have red cells which contain both normal hemoglobin and hemoglobin S and are said to show sickle cell trait. Normally, their red cells are able to transport oxygen, but under conditions of the oxygen shortage, the red cells collapse into the sickle-shaped characteristics of the disease. A very interesting observation is that the distribution of the sickle cell allele confers resistance to the distribution of the malarial parasite, the Plasmodium falciparum, and it has been shown that the sickle cell gene confers resistance to the disease, possibly because the infected red cells become sickled and are removed by the phagocytosis before the parasitic reach maturity in areas where malaria is endemic, and the heterozygous individuals are at an advantage compared with the homozygous SS individuals, so that the sickle cell allele is maintained at a high level in spite of its disadvantage in the homozygous condition. Selection in favor of heterozygous individual is called the heterozygous advantage and is thought to be widespread in the natural population and is believed to account for the large proportion of the polymorphism which is observed. Where we can see it in the ratios of 15 to 20% in the Central African area and the areas adjacent to Red Sea in African continent, and then of course we can see it in Far East Asia, Southeast Asia, and of course India, South Asia, and that's how it moves along.